Okay, so 4.5, getting into the mitochondria and the chloroplasts. So it's more of, it's a shorter section, and it's really kind of getting into the structure of the mitochondria and the chloroplasts, because we'll talk more specifically later on here about cellular respiration and then photosynthesis. But yeah, the mitochondria, we call that the powerhouse of the cell when you're like in middle school, and that's because it makes the energy for the cell. Specifically, it's going to make ATP. So ATP, you'll hear it called like the energy currency of the cell, or basically ATP is this little molecule that can kind of act like a battery, where you can make ATP, send that little battery to your other cells, and those cells will use ATP to do a bunch of different kinds of stuff to like help with running chemical reactions, flexing muscles, whatever it might be. And then chloroplasts, that's where we're going to do, and we call that process cellular respiration. So cellular, it's happening in the cell. Respiration, like if you're respirating, you're taking in oxygen. This process required to make ATP, you use oxygen to do that process. Anyways, then you have photosynthesis, so plants and then some algae will be able to do photosynthesis. So photo, it's using light to synthesize or make glucose. So glucose then gets put into the mitochondria, if you're in a plant cell, to do cellular respiration. So in other words, a common mistake people make with the chloroplast is they think that the chloroplast makes energy for the plant cell. It doesn't make energy, it just gives it glucose. How it makes energy, by energy I'm talking about ATP, is still in the mitochondria through cellular respiration, just like animal cells would do, or other cells that don't have chloroplasts. And then kind of a little more randomly, we'll talk about is the peroxisome. Basically, the peroxisome is just used to, it makes hydrogen peroxide. So peroxisome, hydrogen peroxide. That's the main thing to kind of to understand. And essentially, we'll make these things called free radicals in the cell. So just through certain chemical reactions, these free radicals get made, and then we can take those free radicals, put them in the peroxisome, where they'll make hydrogen peroxide to kind of neutralize those free radicals. So anyways, getting into you're hanging out with your friends, and you're like, wait a minute, where did the mitochondria and the chloroplast come from? And this is interesting because if you look at a mitochondria and the chloroplast, you'll notice that they both are made of a double membrane. And you know that cells are surrounded by a double membrane. Okay, but there are a lot of things in the cell, like the Golgi apparatus, the ER, that they're made of a double membrane. They contain ribosomes, and then they have circular DNA molecules. Meaning, the mitochondria and the chloroplasts, they have their own set of ribosomes. So the proteins that are used in the mitochondria and the chloroplasts are all made in-house. They're made in the ribosomes. And they have, which is why they then have their own DNA. They have their own genes to make their own proteins. And they can grow and reproduce independently. So when a cell needs to divide, or if we need to make more mitochondria or chloroplasts in a cell, they just do that themselves. And so that's kind of freaky, because you're like, oh, mitochondria and chloroplasts, they sound awfully similar to like actual cells. And that's the idea of the endosymbiont theory, or the theory of endosymbiosis. So basically the idea here is, if I jump to the picture, the idea would be, here's our mitochondria. The idea, like say the mitochondria or say the chloroplast, these were at one point in time prokaryotic cells. They were once these bacteria that existed independently of these eukaryotic cells, doing their own thing. And we had like our ancestral eukaryotic cells. So you have like the endomembrane system we went over in 4.4, and then at some point, just in the evolution of life, a mitochondria got engulfed by phagocytosis into that ancestral eukaryotic cell. And basically, this gave this cell an evolutionary advantage. By having these two cells together, making like a supercell, they could work together. Symbiosis is referring to like two things working together, where this would be an example of mutualism where they both benefited from this arrangement. And then perhaps at some other period of time, this would then be like your modern day animal cell. Okay? And then let's say one of those animal cells, like a subset of those animal cells, then engulfed a photosynthetic prokaryotic cell. So in other words, this photosynthetic prokaryotic cell existed by itself independently, and then it got engulfed by like one of these like non-photosynthetic animal cells. And then similar idea, because they could then work together, you could then have this cell would have an evolutionary advantage over like other cells like that maybe didn't have that, or maybe didn't just like kill off everything else, but basically this cell could then at least like this is a good arrangement, and so this would propagate throughout the population, and this would be your modern day plant cells, or your photosynthetic or not even photosynthetic bacteria, but like just your modern day plant cells. So that's the idea, is that they got engulfed. One thing I forgot to leave out with the circular DNA, that circular DNA is what is found in prokaryotic cells. So eukaryotic cells, our cells, we have the double helix. The DNA is a double helix. Whereas prokaryotic cells, they're going to have like the circular DNA. Or I should say, our chromosomes are arranged in like the X-shaped, whereas the prokaryotic cells are just arranged in a circle, is a better way of saying that. Anyways, just, it's, when you kind of show it there, it's like, it might seem a little like, what? Like, did that really happen? And, I don't know, did or didn't, but that's the best kind of guess we could have for how these things could come about, and sort of the best explanation we could say for how would you explain these three pieces of evidence. Okay? So this is just what I said on the slide with the picture of it. Everything just in text for you. So there you go. Make sure I got everything here. Okay. Anyways, moving on. Getting into like the structure of the mitochondria. So here's the mitochondria. First thing I want you to notice is all of those folds. Remember, I've said it many times now. Those folds increase surface area. Cells want a higher surface area compared to the volume. Basically, we maximize our volume by having those folds. This will give us more space for activities. As these folds, all along the folds there, we can have enzymes, where in the mitochondria's case, those enzymes along the folds can be used to make ATP. If it's the chloroplast, we could use the folds of the chloroplast. You could use those to do photosynthesis. The mitochondria, it's double membrane, so it's going to have an outer membrane and then an inner membrane. So this outer membrane would have its own phospholipid bilayer. If I can draw my, it's awfully drawn, but anyways, you get the point. You'd have the outer membrane has that, and then the inner membrane would have another set of another phospholipid bilayer. Okay, and what that does then is kind of like what I showed you with the nuclear envelope. You then will have an inner membrane. I don't know if I actually have it on this picture, but there would be an intermembrane space here. It's right here. Intermembrane space. A space between the outer membrane and the inner membrane. And then you would have the actual inside of the mitochondria. So when we get into the details of cellular respiration, we can utilize those two spaces to set up a gradient that will then be used to power these proteins that will then be able to make ATP. Okay, so that's like a crucial detail of the mitochondria. It's not just like a random thing that is just there. Notice the circular DNA there, and then you'll have those black or brownish dots. Those represent the free ribosomes. So again, the ribosomes of the mitochondria will make proteins that stay in the mitochondria. And we'll call that the mitochondrial matrix. And then these folds that protrude into the matrix, those are called cristae. So yeah, make sure I got everything here. This bullet point, you know, again, once we get into the actual details of cellular respiration, that will make a little more sense. Okay, cool. So getting into the chloroplast. So let me show you the, let me just hop into the actual structure of the chloroplast. So it's similar to the mitochondria in the sense that it has an outer membrane and an inner membrane, and then you have that inner membrane space. So a similar thing can happen where you have an inner membrane space, and then you'll have an inner part of the chloroplast, where we'll call that the stroma. So basically, we can utilize the stroma, that interior and the inner membrane space, use that to set up a gradient that can then be used to make glucose. Anyways, part of the structure of the chloroplast, you'll see, well, first off, you see all these folds here. And again, those folds increase the surface area compared to the volume. And it's kind of interesting because you'll have this stack. So this is like the stack of, there's little, they call them poker chip things. This whole stack is called a granum. The stack of poker chips is the granum. Each individual poker chip, excuse me, each individual poker chip is a thylakoid. Here's how I remember it. The stack of poker chips, it's like a fat stack, like a grand, it's a nice big old stack of poker chips. And then thylakoid, I don't know, it's just one of them. It's just the other one, because I think it's hard to remember which one's the thylakoid, which one's the granum. So if you can keep one of them straight, hopefully you can remember the other one. Yeah, it's I think that's about it. Let me go back to these slides. Oh yeah, chloroplasts will look green because of this green pigment called chlorophyll. Not to be confused with borophyll, but chlorophyll is kind of one of that key pigment molecule that will absorb light. That'll be absorbing the light, and it'll be used to do photosynthesis. The reason why chlorophyll and all plants will look green is because the chlorophyll pigment reflects that green light. Remember how you see colors is by what is reflected, the color of light that's reflected back to you. Yeah. Found in leaves, and then basically anything that is green in a plant, generally speaking, anything that's green can do photosynthesis. So like the bark of a tree, the tree trunk doesn't do photosynthesis because it's not green. It's not green because it doesn't have chlorophyll. It doesn't have chlorophyll, it can't do photosynthesis. Okay. Yeah, thylakoid, random stroma. Yeah, and chloroplasts are part of this other group called the plastids. Talked about plastids a little bit when we were doing in Unit 1, looking at like the storage, like starch, the storage of glucose. Okay, so anyways, getting into the peroxisomes. So with peroxisomes, the main thing to really get at is hydrogen peroxide. It's not like a really heavily tested organelle, but peroxide, hydrogen peroxide. So we make that hydrogen peroxide, and then we'll then want to convert it back to water because hydrogen peroxide itself is that's in like that brown bottle, and sometimes people will pour it on like cuts to help kill bacteria or whatever. But yeah, a big function of peroxisomes is in detoxification of alcohol. So in the liver, along with the smoothie you would find a lot of peroxisomes if you drink a lot of alcohol, because you're going to want to have some way of detoxifying that alcohol. Anyways, we don't really know how they came about in evolution. I don't know, it's kind of a random bullet point, but if you're curious, like, where do they come from? So yeah, I think here's a picture of it. I don't know, it's just a vesicle, just like a lipid bilayer sac that's got a bunch of enzymes in there that makes the hydrogen peroxide. Okay, well, I'll go and end that video there just for 4.5.